Hello, everyone, and welcome to CF Gene's Building Blocks for You and Your Family. My name is Kayler Young, and I have 2 children with cystic fibrosis. My CF journey began August 8th, 2019, after receiving a phone call from our pediatrician that our beautiful daughter Evelyn had an unusual flag on her newborn screen. He went on to tell me that she had a gene mutation for a genetic disease called cystic fibrosis. At that time, I knew very little about CF genetic testing or that my baby even had a newborn test done in the hospital. Therefore, I am happy to be moderating the session today so we can walk through the basics of CF genetics, newborn screening, and genetic counseling together. I'll be introducing our other speakers in just a moment. Before I do, I wanted to provide a few housekeeping items. This session is being recorded and will be available after the event on the CF Foundation YouTube channel. You'll be notified by email when session recordings are available. After our presentation, we will take a quick 5-minute break and we'll be hosting a 45-minute extended Q&A with today's speakers. So please hold all of your questions until then. We won't be able to answer questions about individual diagnoses or treatment plans, and we encourage you to use this information you learned today to have conversations with your care team. The extended Q&A will take place in the same virtual room at 9:15 p.m. Eastern Time. The Q&A portion of this program will not be recorded. If you experience any technology problems during the session, please visit the help desk on the left-hand side of your screen. We also want to remind everyone of our community guidelines. Please be kind, open-minded, and respectful of everyone's opinions and life experiences. And please do not ask or give medical advice. If you need staff support at any time or want to report a violation, we also encourage you to visit the help desk. There will be a few polling questions from us panelists, which you'll be prompted to answer throughout the session. There is actually one live right now. These polling questions will help us get an idea of who everyone is and your familiarity with the various topics covered today. Polls can be found on the right-hand side of the screen next to the Q&A and chat. With all that out of the way, let's introduce our panelists. Welcome, Dr. Richard Parad, Associate Professor of Pediatrics at Harvard Medical School and Director of Newborn Genomic Medicine Program at Brigham and Women's Hospital in Boston, and Kimberly Foil, Certified Genetic Counselor at the Medical University of South Carolina. We're so grateful to have your expertise here with us today. My son Bennett was born in 2017. At that time, his birth and infancy were typical, and there were no red flags in his health history that would have indicated any issues. When he was around a year old, we noticed when he would get a cold, the cough would linger longer than what we thought was normal. At the peak of one of his illnesses, he was diagnosed with pneumonia. His pediatrician at that time didn't have any concerns, and we credited a developing immune system as a reason for the sickness. After getting a phone call about my daughter's newborn screen, we immediately scheduled a sweat test at our children's hospital to confirm her diagnosis. Upon arrival, I was told that they often test siblings as well, but that we could wait until a later time if we wanted. I knew right away I wanted to have Bennett tested along with his sister, as it would always linger in the back of my head if he had CF as well. Within an hour after the testing, the genetic counselor accompanied our new CF clinician, and they told us both children's sweat chloride tests fell within the low 60s range, which was indicative of cystic fibrosis. Dr. Prad, as our family tried to learn more about CF post-diagnosis, one thing that remains a little confusing is the genetic terms, and what exactly is a mutation?
Well, thank you for that question, Kayler. I'd like to go through some background for the listeners on the building blocks that we talked about in our title so that perhaps they can understand CF genetics a bit better. We're going to go back to high school biology and bear with me as I define some basic terms for you. Over on the left of the slide here is a cell and a nucleus in the center of the cell. All the organs in our bodies are made up of these cells, and every cell has a nucleus. And inside of each nucleus is a blueprint or instructions for making all the proteins that the cell and the organs and the body need to function. There are 23 chromosomes, which are packages of DNA that contain the information in that blueprint. And each cell, in almost all the cells in the body, each cell's nucleus has 2 sets of these 23 chromosomes. You can see over here on the right, if I pull out chromosome number 7, there is a short and a long arm, they're called, to this chromosome, and a little band on the longer Q arm of chromosome 7 is where the gene responsible for cystic fibrosis, the CFTR, or cystic fibrosis transmembrane regulator, conductance regulator gene, is located. We go back to the diagram on the left. You can see that these chromosomes, if you stretch them out, are made of a highly coiled tiny strand of DNA, very, very long, and spaced along this strand of DNA are blocks called genes. And each gene is made up of a sequence of chemicals called nucleotides. There are 4 different types of nucleotides. Let's call them A and T and G and C. There are two strands coiled in a double helix in this long strand of DNA. And the A is usually matched with the T and the G with a C. And the order of these nucleotides, or base pairs as they're called, is what provides the information to code for the synthesis of proteins. So as I said, each cell has 2 copies of the 23 chromosomes. On the right here, we're going to represent a mother and a father who are CF carriers, and their blue half is the abnormal CFTR gene that they carry on the chromosome 7 in every cell in their body, and the black side is the normal copy of the CFTR gene. And for this couple, there are 3 combinations of their genes, their CFTR genes, that can happen when they have children. They can get a child who receives 2 copies that are normal. They can have a child who has 1 normal and 1 abnormal copy. They are carriers as themselves. Or the way an affected child is born is if both of the CFTR copies that go to that child carry an abnormal CFTR gene. So what is the protein made by this gene, and how does each cell make this protein? Well, let's go back to our 2 chromosomes now, a slightly different cartoon. Here are the 2 chromosome 7s that live in every cell. And let's take an example of a cell that is in a person who is a carrier and has 1 normal CFTR copy on 1 chromosome 7 and an abnormal one on the other. First, we're going to talk about what happens from the normal chromosome. The DNA, if we stretched out that little band there, the DNA that codes for the CFTR gene is made up of blocks of sequences called exons. These are the little blue boxes, and introns are the little spaces in between them. And inside the nucleus, the machinery in the cell will transcribe or copy this sequence from DNA into a copy of a slightly different chemical called RNA, which then exits the nucleus of the cell into the fluid around the nucleus, where all the components for making proteins are located. And in the process of converting from DNA to RNA, all the intron sequences are cut out and the exon sequences are knitted together. The protein that's made out in the cytoplasm, the liquid around the nucleus, the protein for CFTR is about almost 1,500 amino acids long. That's a moderate-sized protein. And here are 2 pictures of what the protein looks like. The cartoon on the left shows the basic structural parts of the protein. The transmembrane domains here in blue and green, these are the parts of the protein that weave in and out of the cell membrane. So this protein lives on the surface of the cell. And you might imagine there's a little hole here. This is a pore that exists in the middle of these protein components. And then there are three parts that live inside the cell, and they are called nucleotide binding domain 1 and 2 and the R domain. And these are involved in regulating the opening and closing of the hole in the protein. So this picture on the right is really much prettier and more accurate of what the protein looks like sitting in the cell membrane. And there are many, many copies of this protein sitting in the membranes of cells that make this protein. Not all proteins end up turning on production of this protein, but we'll talk about where it is made. From the other chromosome, let's take an example of a chromosome that has the most common variant or mutation found in the CFTR gene. It's called F508 delta. There are 3 nucleotides missing from the normal sequence, and that's going to be represented as an X here. So those missing nucleotides are transcribed into the RNA copy that's going to make the protein. So again, it's missing 3 base pairs. Those 3 base pairs happen to code for 1 amino acid named phenylalanine, so that the final protein is going to miss in this nucleotide binding domain a phenylalanine amino acid. And that's all it takes to potentially sort of mess the protein's function up. So over here, you can also see the X, that this mutation location is on the inside of the cell and in the part of the protein that, although it is the part of the protein that controls the opening and closing of the channel, it actually causes problems because it doesn't allow the protein to fold into this shape it needs to fold into to insert into the cell membrane. Let's talk a little bit more about what this protein does. As I said, it's a chloride channel. And in this cartoon, the green layer here is a mucus lining. In this picture, we're talking about an airway in the lung and a cell in the airway, either an ionocyte or an epithelial cell. And the protein is inserted in the cell membrane. If it functions normally, this part of the protein opens up and allows chloride ions to travel from the inside to the outside of the cell. When that happens, when salt moves in and out of the cell, water also moves out into the mucus, and it keeps that mucus layer flowing and liquid. If there's not enough water in that layer, it gets very thick, and that's when problems start happening. This is from the CF Foundation, a video I wanted to show you, another representation of the CFTR protein sitting in a cell membrane. In this case, this is a protein that has a variant called G551D, and because of that variant, the whole protein is made, but the part of the protein that controls opening and closing of the pore or the gate doesn't work properly. You will see flying once I set this in motion, a little green dot here, which is a drug which has been developed, which helps the protein that isn't functioning have some function. And then you'll see these little green dots. These are chloride ions that are trying to get out of the channel to the outside. And you can also see where they bounce off the inside wall of the cell membrane where there is no channel. So let me set this going and let's see if this works. There you go. So no chlorides getting through the channel. The drug comes, the protein opens up. Suddenly the chloride can get through. That's normal function. And chloride and water can move to where they need to go, and the mucus layer is working well. Okay, so just to review how this protein is made, the DNA in the nucleus If there's a normal sequence, makes a normal RNA copy that leaves the nucleus, goes into the cytoplasm, starts making the code for the appropriate amino acids, cause them to be linked together using ribosomes, and the protein is processed and travels to the surface of the cell where it functions as a chloride channel and allows chloride and water to move in and out of the cell. If this protein doesn't work, once again, and the ions in the water can't move the way they need to, then the mucus layer gets very thick and it can't perform its functions. So how does a change in CFTR affect the body? We've been talking about that mucus layer. Why are we talking about that? Well, the thing that ties together all the organs that seem to be most involved in cystic fibrosis are organs that carry tubes or pipes in them. And there are many organs that do that. The lungs have airways, those are tubes. The pancreas has a big duct and many little ducts in it, as does the liver. The intestine is a big tube. The little sweat glands on the skin are all tubes. If the CFTR gene has a defect and the protein is abnormal, and that mucus layer doesn't have enough water, it is very thick and it will plug up those tubes. And when the tubes get plugged up, things back up and things get injured behind the obstruction. So it's this common occurrence that is why all the organs are affected in that way. And then lastly, about CFTRs, why do different CF patients have different severities of CF? And it turns out that although we can't always predict with certainty exactly how severe the CF will be based on the mutations that we identify right at the beginning, we do know that certain mutations tend to act in certain ways, sometimes more mildly or with symptoms presenting later on. And we'll talk about what we know about that. So let's go back to DNA and a cartoon of DNA, the gene, the DNA bases, the nucleotides that make up the gene, that sequence that is coding for the amino acids. And use the analogy of the DNA being like a sentence, where each word in the sentence is equivalent to a gene, and each letter in the gene is equivalent to one of these nucleotides or base pair letters. You can see if I change the gray cat ran down the hall, if I change one letter from hall to ball, the meaning of the sentence completely changes. The gray cat ran down the ball, that means something else. And this is just an analogy of what changing DNA sequence can do to the way proteins work. But don't get overwhelmed by all these colors. I'm going to talk you through this. I'd like to actually first draw your attention to the upper right corner here. And if we look at the top line as a DNA sequence, you can see in this cartoon that the nucleotide letters have been broken up into little groups of 3. That's because these are called codons, and each 3 base pairs code for an different amino acid. And this is the secret decoder ring down here that the biochemist can use to say if it's CAC, that codes for the amino acid valine. There are about 21 different amino acids that get tied together to make proteins. Now, for this particular protein, if there is a variation in the sequence or a mutation at this site, the CTC, which normally codes for glutamic acid, if that T is changed to an A, it's a different amino acid that gets put into the protein, valine. And that little change is enough to cause a disease. So let's go back over to the left and talk about different types of base pair replacements. If the top here is a normal sequence, and we look at this red T here, what's called a base substitution would be if the DNA sequence has been changed because for some reason, instead of that T being there, there's a C present. So you've substituted one base pair for a different one that normally shouldn't be there. Similarly, you could imagine sticking an extra base pair in, so that can happen as well, or losing a base pair, or in the case of F508 del, there are 3 base pairs that were lost, and that caused the mutation that causes CF in many patients. Now, this is called a missense mutation if one base pair is substituted for another. Missense mutation. The same number of amino acids, but one has been swapped out for a different one. If that happens and something called a stop codon is created, so instead of changing to another amino acid, it's a code for stop that tells the cell machinery to stop making the protein. That's a severe mutation because the full protein can't be made, and there's not the same hope that at least some function might be there if the whole protein could go on and get finished. So that's called a nonsense mutation if a stop is inserted. And missense and nonsense mutations are a type of way of labeling the different variants in CF. And typically nonsense mutations are more severe and may cause more severe disease. And if you're lucky with a missense mutation, if there's not a very big difference in the amino acid change, then the protein may still have some function. Lastly, in the CF world, a special category for CF variants has been created. They're classes 1 through 5. I'm going to quickly walk you through why those classes are different. Classes 1 through 3 tend to cause more severe CF, and classes 4 and 5 tend to cause milder CF. The vast majority of patients have class 2 variants because delta F508 or F508 del falls into this category, and that's the most common variant among CF patients. If we look at class 1 and compare what happens in the cell to normal physiology, the DNA, remember, makes the RNA makes the protein, and then the protein is processed, put up to the cell surface, and functions as this little channel. In class 1, if there is a stop created by a change in the base pair sequence, then no protein gets made that can go to the surface, and there's no protein there to work, and that's a severe problem. And that was actually that G542X variant that you're going to hear about in Kayler's husband falls into this category. Class 2, again, the most common one, is a change in the protein where the protein is also whatever variation has happened in the amino acid sequence causes the protein to not fold properly or not be processed properly. It can't get to the surface. If it's a class 3 variant, a full protein is made, but there's a variant in a place where the control of the opening and closing of the pore takes place. And so the protein is there, but it can't really do anything. And this is a protein that where many drugs that are now very effective in CF can work, where it's been figured out how to add a molecule to get that protein to have some function. Class 4 also makes it to the cell surface, but this often may be a part of the protein that is embedded in the cell membrane, and it may crumple up the pore or make it not be as open as it should be, but there is function to the protein. It's just diminished function. And then in the last class, normal protein is made, but because of the regulation of how the protein is made, only a tiny amount of normal protein is made, and that can also cause symptoms. So to put this all together, if we look at sweat chloride as a measure of how well the CFTR protein is functioning, and if we say a normal person who has 2 normal CF genes has 100% normal CFTR protein function, their sweat chloride would normally be in this range below 20. And here green is the newborn and orange is adults. If you have, say, two class 1 through 3 variants, those are the most severely affecting the protein, then there may be no function or very little function, and that's when the sweat chloride is high. And any value above 60 millimolar per liter is considered diagnostic of CF. And then we have some categories in the middle. Carriers tend to have about 50% function, but that's plenty. That's enough function for there to generally not be symptoms. And then there are some groups in between that we have a little trouble with that don't present with very classic symptoms.
Kayler, you're on mute.
Sorry about that. Thank you, Dr. Parad. So in our experience, both of our children were taken at birth to be tested, but we were never told they were to be tested and how the testing was done. So when our daughter's screen came back positive, it was a bit shocking as we didn't even know that they were testing for CF. Can you go into a bit more detail on what newborn screening is and how that works?
Certainly, Kayler, and I think it's very common. There's a lot going on when a new baby joins the family. Parents are overwhelmed with a lot of things. They don't necessarily read everything that's put in front of them, but most states do provide some kind of pamphlet or information about newborn screening, both before the baby's born and before the baby goes home from the hospital. And then it either isn't seen or the parents forget that anyone told them. So let me tell you a little bit about the background for newborn screening in general. Newborn screening started about 60 years ago in Massachusetts, my state. It was the first state to screen for a rare metabolic disorder called phenylketonuria. And that's because this man, Robert Guthrie, who was a pediatrician in New York in the late 1950s, had developed a test that could be used on, done very quickly on newborns that could diagnose this test from just one drop of blood. More importantly, he created a system, and it led to the development of a public health infrastructure that allowed for the collection of that drop or several drops of blood on every newborn to go to a centralized state laboratory rapidly after the baby's birth, and to be able to test or screen for a number of disorders that where the baby would look to have no symptoms at birth, but symptoms would gradually develop. And if there were therapies available for those disorders and very early initiation of those therapies, what impacted the baby's outcome, the order could be considered also to take advantage of this infrastructure. So the infrastructure includes the use of this card. It's named the Guthrie card after this man, which has information on the baby and the baby's pediatrician, so the results can go to somebody, and then some blood collected in these little circles on the filter paper. So there are about 4 million babies born in the US each year. About 1 in 300 are found to have one of the disorders that are looked for in newborn screening. And there are 3 different types of newborn screening. There's this heel stick filter paper blood group of disorders I just described to you. There's also hearing screening and congenital heart disease screening. Currently, most states, now each of the 50 states has their own newborn screening program and their own panel of disorders that they screen for in their own laboratories. Currently, on average, about 36 disorders are screened for based on some government standards that have been developed, but that varies in each state. The menu is slightly different in each state. And for cystic fibrosis, from the CF registry, we know in 2021, about 800 new patients were diagnosed. And also from the registry data, we know at least 75% of those patients were in 2021 were diagnosed through newborn screening. And we'll get at the end to how that compares to years past. So this process of newborn screening includes testing for a number of disorders that must be treatable, must have an available test, and where early detection is critical. The dry blood spots are collected, and this is a universal program. So every baby is supposed to get this. It's not something you pay for out of pocket. It's just supposed to happen in your hospital, and your hospital orchestrates this. The family's supposed to be informed. The laboratory responsible for the testing has very high assurance of quality and consistent lab results. I'm sorry, my slides keep moving ahead without me doing anything here. And the result reporting is also critical. So it's 4 million babies that are having a lot of tests done, and then the information on the results has to get to the pediatrician, who should then transmit the information to the family. Now, the history of CF newborn screening goes back, I'm going to just talk about 4 stages in this history. It goes back to the country New Zealand, where in the late 1970s, Jeanette Crossley made the observation that newborns with CF had an elevation in their blood of an enzyme called trypsinogen. This is an enzyme made in the pancreas, and even in the fetus, before the baby is born, in an CF-affected fetus, the pancreas is already having trouble because that tube that runs through the duct that runs through the middle of the pancreas gets plugged up with thick secretions, and that actually backs up the enzyme into the bloodstream, and the blood levels are elevated. So she noticed that on a dried blood spot you could get from a heel stick, you could measure the IRT. And the problem was, though, that CF wasn't the only thing that caused IRT to be elevated. There were sometimes just problems in a difficult birth that transiently increased the IRT level. So she noticed that if you check the level again, maybe 2 to 6 weeks later, if the level stayed up, then CF was the likely diagnosis. But if it went back in the normal range, then that was probably an initial false positive. So this is called the IRT because there are it's measured 2 times test for CF. It started in New Zealand. In the United States, the first state to think about using this was Colorado. They did a pilot in the early 1980s. And then Wisconsin was the next state to do this. They did a huge research project looking at randomizing 750,000 babies born in the state of Wisconsin to either receiving CF newborn screening and identifying the baby very early, or letting them present when they normally would present, which was at about, on average, about 4 years of age. And then they followed these children over decades and showed that just by knowing the baby had CF and jumping on top of nutritional interventions, which was all that was mainly available when the project started, that those babies had better growth of height and weight, and their lung function was better maintained, and they did better. And it was these data that led the Center for Disease Control to call together a conference in the early 2000s to try to convince states that this was a disorder that they should add to their newborn screening panel. Initially, it was the IRT test, but as more and more people studied newborn screening for CF, different algorithms developed. And the most important initial change was, instead of doing that second IRT, DNA was tested from that same initial dried blood spot, because technologies had been evolving for being able to look for mutations in tiny DNA samples. The very first algorithm just looked for delta F508 or F508del, the most common variant, but there are over 2,000 variants reported in this gene. And so that wasn't going to pick up everything. And in Massachusetts in 2004, we reported a 4-year experience of using a multi-mutation or multivariant screen, where we looked for the 12 most common CF variants. And this has expanded to the point where some states actually will sequence the entire CF gene as a second or third test after an elevated IRT. So in our algorithm, we would take the babies with very high IRTs, look at their DNA, and then if they had any DNA variants noted, we referred them for a sweat test, and that would make the diagnosis. Sometimes there were babies that had very, very, very high IRT, but we didn't find any variants, but we thought, well, maybe it's one we're not testing for. So we would send those babies in for a sweat test too. And anything that was below the cutoff for the IRT at the beginning, those babies were reported out as being normal. We also noted, though, with our multivariant panel, that we picked up a new problem, one that we weren't really looking for, and we still haven't completely solved. But you may have heard of this term CRMS, or CFTR-related metabolic syndrome, which in Europe is called cystic fibrosis screen-positive inconclusive diagnosis, or CF-SPID. And these are cases where the IRT is elevated. So this term should only be applied if the baby has had a newborn screen done and the IRT was elevated, and then either not too clearly pathogenic CF mutations are identified, but at least one, or when the baby comes in for the sweat chloride test, there's one mutation, but the sweat chloride isn't above 60. It's in a sort of intermediate zone. And so these are babies that I like to call this label. It's not really a diagnosis, but a sort of categorization as like putting someone in a parking lot. And then you have to watch and wait to see where you're going to go out of the parking lot to. You're either going to end up not having CF and maybe just being a carrier, or you may have CFTR-related disease, which is not sort of full-blown CF, or your sweat chloride may go up over time and you actually do have CF, and this is just how your variants have behaved. So how does CF newborn screening impact CF patients? What's the good that CF newborn screening has done? Well, it's dramatically decreased the age at which the diagnosis is made. There used to be very lengthy diagnostic odysseys in identifying CF patients. We see much less of that now. That allows for earlier initiation of therapies, and there are many more therapies than there were even 10 years ago. So, and all these things have made a difference in improving the outcome and the long-term survival of children and adults who have CF. So as I noted before, in the past, before newborn screening, the way babies would present to their physicians would be at an average of about 4 years of age with symptoms of malnutrition and recurrent respiratory infections. And this is a baby with a sort of big bloated belly with something called kwashiorkor or protein malnutrition because of the malabsorption diarrhea and not being able to be well nourished. Now we have newborn screening, and the vast majority, over 75% of babies, are diagnosed looking entirely healthy, and we're not kind of starting behind the 8 ball. We're starting with a fairly healthy looking baby to get the awareness that there's going to be trouble if we don't watch for things and start therapies early. So then the average age of diagnosis was somewhere around 4, and now half of babies have a definitive diagnosis by about a month of age. And that's allowed for us to institute therapies earlier. 2010 was when all 50 states had adopted CF newborn screening. Now I can't say that the median survival age going up from 30 to 50 can be totally accounted for by newborn screening, because lots of other things have happened since 2010, like a lot of drugs that have developed. But newborn screening is one factor that has been involved in this uptick in the curve of survival. Finally, the CF Foundation did publish within the last 2 years, using their patient registry, a look at how infants had been doing since newborn screening was universal in the United States. And this has pointed out to them, well, things are better, but we could do better. And there are areas where we still need to improve both the infrastructure and what we look for in our variant panels. Finally, I'm sorry. We have also worked on developing some consensus guidelines for better newborn screening, more consistent across the 50 states. The CF Foundation has an initiative that has been going on for over 10 years, where they provide grants to states to look for better ways, more timely ways, more equitable ways of doing their screening. And they've been trying to connect newborn screening state programs better with CF centers to work together, and also to improve the panels so that identifications of variants that occur in minority populations that are different from the more common Caucasian populations are not overlooked, and there's equity in screening and identification, so therapy can be started.
Thank you so much, Dr. Prad, for that great overview on the genes for CF and explaining some of the most common questions people have when dealing with a new diagnosis. Before we move on to the next portion, please take a moment to answer the quick poll on genetic counseling. And now we'd like to turn the discussion over to Kimberly Foil to share what happens after diagnosis.
Thank you, Kayler. So I'm a genetic counselor embedded in the pediatric and adult CF care teams. And to give you a little bit of background about what is genetic counseling is the process of helping people understand and adapt to genetic contributions to disease. Genetic counselors review personal and family medical histories to assess the risk of a condition or facilitate diagnosis of a condition like cystic fibrosis. And we use that genetic information to guide personalized care and optimize outcomes for the patient and family. Genetic counselors are master's trained, board certified professionals with expertise in both medical genetics and psychosocial counseling. But what are the CF recommendations for when genetic counseling should be offered to patients and families? Next slide. In 2008, diagnostic guidelines recommended that families of infants diagnosed with CF should receive appropriate education at the first diagnostic visit and that genetic counseling should be provided. Additionally, recognizing the familial component, a sweat test should be performed on siblings of somebody with CF. Later guidelines indicated that genetic counseling should be offered to families of individuals followed for CFTR-related metabolic syndrome, including a discussion of the risk for future pregnancies. And most recently, consensus guidelines affirmed that genetic counseling is recommended for all parents of infants who have a positive newborn screen for cystic fibrosis. Next slide. What is happening in actual practice? There's a lot of practice variation for a number of reasons, and a survey conducted found that nearly 40% of CF center directors or clinic coordinators never refer newborn screen positive infants to genetic counselors. My group conducted a study in 2018 that also looked at utilization of genetic counseling both in pediatric and adult settings. And we found that just over half of newly diagnosed infants or children received genetic counseling in a CF center visit, where some others received genetic counseling by referral or situationally based on things like whether the parents plan to have additional children. Whereas in the context of an adult diagnosis, a minority of patients received genetic counseling in the CF clinic, and many rarely or never received genetic counseling at all. Next slide. Dr. Parad gave us a fantastic overview of CF genetics, and understanding this is really the bread and butter of what genetic counselors do and help convey to families so that families can make meaning of a diagnosis and integrate it into their healthcare decisions. So genes, of course, are units of our DNA, and they're our body's blueprint or recipe for making proteins like the CFTR protein. We all have 2 copies of the CF gene, one that's inherited from our mom and one that's inherited from our dad. And when we have children, we each pass on one of our 2 copies. So most people have 2 normal copies of the CF gene. Carriers have 1 normal and 1 abnormal. And people with CF have 2 abnormal copies or 2 faulty instruction manuals for making the CFTR protein. Variants or mutations in the gene change the CFTR protein product in a variety of ways, and over 2,000 different mutations or spelling changes in the DNA sequence of the CFTR gene have been identified. These changes then result in either not enough CFTR protein and or dysfunctional CFTR protein being produced. And a person's total and type of CFTR protein depends on both gene copies or both alleles, which together make up what we call the genotype, or which type of CF genes do you have. Knowing a person's genotype is important in confirming the diagnosis, as well as offers some what we call genotype to phenotype correlation, or what do we expect for people who have that combination of mutations. It also enables accurate family testing and, very importantly, guides treatment decisions. Next slide. With over 2,000 mutations in the CF gene, it's been really helpful to bin these mutations into functional classes, where our class 1 mutations are variants that result in no CFTR protein being produced. Class 2 mutations result in production of an abnormal CF protein, but the cell discards it and it doesn't reach the surface of the cell or the membrane where it's needed to function. Class 3 variants produce a protein that reaches the surface, but it remains stuck in the closed position rather than opening to conduct chloride ions as necessary to regulate that fluid balance inside and outside of the cell. In an untreated or unmodulated state, the class 1 through 3 mutations generally result in classic severe cystic fibrosis with pancreatic insufficiency. Classes 4 and 5 are often termed residual function variants, and as that term implies, there is some remaining function of the CFTR protein related to class 4 and 5 variants. They are CF-causing variants, but people who have at least one class 4 or class 5 allele are often pancreatic sufficient and don't need to take pancreatic enzyme replacement therapy. On the right-hand side of the screen, I have another schematic that refers to overall CFTR function related to the degree of symptoms. An individual who has 2 normal CF genes would have 100% CFTR function, be normal, not a carrier. Our carriers with 1 normal gene and 1 abnormal generally have around 50% of CFTR function, which is adequate to protect against CF symptoms in most individuals. As we move lower down in terms of our CFTR function, we hit the gray zone of CFTR-related disorder. This may occur in people who have very mild CFTR variants. CFTR-related disorder can be a variable entity, oftentimes causing symptoms in a single organ or system, and those symptoms may be milder or present later in the lifetime. We've even diagnosed some adults, say, in the 6th decade of life, who have a presentation that benefits from CF treatment but doesn't meet the picture of classic cystic fibrosis. As we drop down to about 10% of CFTR function, we see cystic fibrosis with sweat chlorides in the diagnostic range, but oftentimes pancreatic function intact. And as we move down to about 3% or less of CFTR function, the pancreatic function is knocked out. And most of the CF community actually falls into that category of having 2 severe mutations resulting in pancreatic insufficiency and the multi-system disease we think of as cystic fibrosis. The next slide. One of the tools that I like to use as I'm preparing to see a patient, and oftentimes when I'm with the patient as well, I'll bring this up, is the CFTR2 database. And this is a database where you can type in the name of one or two mutations to see the significance of those variants, either alone or in combination. The CFTR group researches in cellular models the impacts of various mutations to determine whether they affect CFTR protein quantity, conductance, or both. And this database is also interfaced with the CF Foundation patient registry, such that in a de-identified fashion, it can pull numbers of how many patients are living with this CF mutation, and on average, what sort of health parameters do we see, such as average sweat chloride for people with the genotype, lung function, and importantly, percent with pancreatic insufficiency. So in this slide, I have put the mutations in that occur in Kayler's children to make this relatable to the case we've been learning about this evening. And we see that this variant combination does cause CF, but that patients who have this variant combination are likely to be pancreatic sufficient. And in the case of this particular genotype, there are just 4 patients in the database with this genotype. But for some genotypes, like 2 copies of the Delta F508 mutation, there are many thousands of people in the database with the genotype, and then the health outcomes data becomes more robust. This site will also give some insight as to whether the mutations an individual has qualify for targeted therapies or genotype-directed therapies. The last decade has revolutionized CF care with the approval of CFTR modulators that treat the underlying cause of CF for many individuals with cystic fibrosis. In 2012, the first compound, ivacaftor, Kalydeco, was approved for a single mutation that results in a gating problem, or a class 3 mutation, where the small molecule therapy acts like a molecular key in a lock to open up that channel and help it work more closely to normal. Several years later, additional compounds were developed, such as Orkambi and Symdeko, for individuals who have 2 copies of the Delta F508 mutation, or in the case of Symdeko, select other mutations. The real breakthrough for the community occurred in 2019 with the approval of Trikafta, a highly effective modulator therapy that provides dramatic benefit for people with at least 1 copy of Delta F508 or select other approved mutations. And these highly effective modulator therapies offer significant improvement in lung function, reduction in pulmonary exacerbations, reduction in sweat chloride, and improved nutritional status and quality of life, among other benefits. And just last night, this slide said that Trikafta was approved for ages 6 and up, but today was a fantastic day for the CF community, as the FDA has now approved Trikafta for the age group of 2 to 5. So this is tremendous, and we're so thrilled to see these modulators that really revolutionize care being available to more and more individuals. But we recognize that not all individuals in the CF community can benefit from modulator therapy. About 10% of people with CF have 2 mutations that do not respond to modulators. And these mutations oftentimes fall into the class that produces no CFTR protein at all. If there's no CFTR protein produced in the cell, then there's nothing to fix with this sort of small molecule therapy. But the genotype is still incredibly important because it opens up the option to participate in research studies, which result in drug development and eventually will bring these sorts of treatments for all with CF. The next slide. The CF Foundation is highly committed to finding effective therapies for all people with CF. For people with class 1 mutations or stop or nonsense mutations that result in the premature stop signal and no CFTR protein being made, many compounds are being screened to potentially read through the stop signal so that a protein could be created and acted on by a modulator. Additionally, early studies of inhaled mRNA and inhaled gene therapy or inhaled gene delivery are underway, and these treatments would result in lung cells expressing normal CFTR protein regardless of a person's mutations. And other strategies such as gene editing are also being looked at, as well as next generation modulators to increase effectiveness and reduce side effects.
Thank you so much, Kim, for that great intro into genetic counseling. One thing I wanted to ask about is the family situation. It was obviously a bit shocking to have our child diagnosed and getting the rest of the family tested to find out our first child had CF as well, and my husband was potentially impacted also. When you get a newly diagnosed adult or a parent of a child into your clinic, how do you go about getting all the answers for them?
Absolutely, Kayler. So every family, every situation is unique. And I go into the session with the background information like I just talked through, and we'll provide some education to the family about that autosomal recessive inheritance pattern, knowing that the parents of someone who's been diagnosed are what we consider obligate carriers, or at least carriers for CF. And so there'd be a recurrence risk of 1 in 4 or 25% should two carrier parents have additional children in the future. Also, if there are existing siblings, we know those siblings are at risk. In the adult setting, I also work with adults with CF who are embarking on their own family planning, as many adults with CF want to have children of their own, as life expectancy and quality of life have improved so dramatically. Most women with CF are able to get pregnant and safely carry a pregnancy, while most men with CF face infertility due to congenital absence of the vas deferens due to the underlying CFTR defects. So working with families, I assess their current situation as well as their goals and talk with them about options to achieve their goals in terms of family planning that may look like natural conception if it's possible, with or without prenatal testing for a CF diagnosis, or if people want to prevent CF from occurring or require fertility assistance based on male infertility, then in vitro fertilization with or without embryo testing are possibilities that we may discuss, in addition to alternative family building models such as adoption or use of a gamete donor. I'd like to make things a little bit more tailored to your family as I talk through some of the other discussions that might occur in clinic. So if we can flip to the next slide. Kayler, I can imagine that if I were to meet you at your first CF center visit, you would have just found out about your daughter's positive newborn screen results. So she would have had an elevated IRT at the state lab, and they would have reflexed to genetic testing and identified the R347H mutation and the G542X mutation. And we know by those mutations that we have a presumptive diagnosis of CF. So of course we'd want to do the sweat test, diagnostic gold standard test to confirm, but we would assume that we are going to have abnormal results based on that genetic result from the newborn screen. And I would want to discuss with you the process of the newborn screening and how we arrived at that diagnosis, what cystic fibrosis means, and how CF happens. I would talk to you about the specific mutations that were identified. Specifically, the G542X produces no CFTR protein and is a severe class 1 mutation, while the R347H is CF causing, but offers residual function. So fortunately, most people with this mutation are pancreatic sufficient. And additionally, people with this variant are eligible for Kalydeco beginning at 4 months of age and later for Symdeko or Trikafta. Genetic counseling usually involves review of the family history. And I'll draw out a pedigree or a 3-generation family tree where we use circles to represent females and squares to represent males, and individuals get shaded in if they're affected with CF and partially shaded if they're a carrier of CF. So in your case, we would have started with your daughter, Evelyn, who was shaded as affected. And I would have learned that you had your son, who, although he had normal newborn screening, hadn't had diagnostic testing because no newborn screen is perfect. I would have recommended, if not already done, that we proceed with diagnostic testing for your son. And in his case, it did turn out that he was affected. We would also offer carrier testing for you and for your husband to determine which mutation was inherited from which parent or occurs on which side of the family. It's very common when I take a CF family history to hear that the CF diagnosis was a surprise or the family history is negative, that neither parent knew of anyone in the family who's ever had CF before. And this is often the case with autosomal recessive conditions because the carrier status doesn't cause problems and can run silently in a family for generations. And there's a risk for recessive conditions to manifest when by chance 2 members of a couple are carriers for the same condition. So in this case, we found that Kayler is a carrier for R347H, and her husband carries the G542X mutation. Oftentimes things are straightforward, and sometimes we get surprises. On her husband's test, we also found that he carries a mild CFTR variant on his other gene copy, and that this gene combination for him actually confers some lifetime risk for CFTR-related disorders. So he can incorporate that into his own healthcare and management going forward. Overall, genetic counseling can offer anticipatory guidance, hope, encouragement, and support to patients and families as they acclimate to this complex information. Next slide. And as we saw in the case of Kayler's family, sibling testing can be really important in picking up individuals who also benefit from proper diagnosis and care. Approaches to sibling testing have been found to vary between pediatric and adult clinics, where it's very commonly recommended that siblings have diagnostic testing when a child or an infant is diagnosed. But when an adult is diagnosed, that recommendation is less common, although important. And there are several examples from my adult clinic where we've diagnosed one adult sibling, gone to the siblings who are also adults and either generally healthy or very mildly symptomatic, and actually confirmed a CF diagnosis that may cause problems 10 years down the road and benefit from the appropriate surveillance and using therapies as indicated, as well as providing that genetic information for the family. Now, most CF clinicians recommend a sweat test as the diagnostic test, but some opt for genetic testing for familial mutations instead or in addition. And a genetic counselor can also help select the right test. It's important to know that a sweat test does not determine carrier status. And in the case of often milder adult diagnoses, the sweat values may be a little bit harder to interpret or more likely to fall in the intermediate range. And in general, any relative of somebody with cystic fibrosis should be offered CF carrier testing as part of their own family planning. Next slide. This is all complicated, and depression and anxiety are common issues in the general population, but have been shown to occur 2 to 3 times more frequently in individuals with cystic fibrosis and their family members. The CF Foundation has done a terrific job implementing guidelines for annual screening for mental health for patients ages 12 and up and for caregivers of children with cystic fibrosis. With a background in counseling, genetic counselors may serve as part of the psychosocial team, along with a CF social worker or psychologist or other provider. Additionally, my center recently collected some not yet published data among parents of infants who had abnormal newborn screens for CF and found that sadness and anxiety acutely rise upon notification of the abnormal screen results and don't return to baseline until after the diagnostic resolution. And in this cohort, most babies were resolved to be unaffected carriers. So this shows us that there's a lot of opportunity to improve the patient experience in adapting and expediting the process of education and resolving infant newborn screens.
Thanks so much for those details. That really helps create a bigger picture for the family to work from. We were lucky that our clinic offered free testing for the parents to confirm our genetics and which parents had which mutations. In addition, we were able to meet with a genetic counselor at our CF center to review the results and the next steps. In our final poll for today, we'd love to hear from the audience on your access to genetic counseling. So please take a second and answer the quick poll on the right. Kimberly, what do you recommend for people if they don't currently have access to a genetic counselor?
Sure, that's a great question. So not all cystic fibrosis care centers have a genetic counselor embedded in the care team or necessarily even at the parent institution. If you don't have access to a genetic counselor through your care center, you can look at the findageneticcounselor.nsgc.org website and search for a genetic counselor who's located geographically close to you or who offers telehealth services. It's important to note that not all genetic counselors have CF expertise, as many practice in other areas such as cancer genetics, but many are familiar with cystic fibrosis. And if you don't have someone that your care team can refer you to, then it's worth taking a look at this website. I'll also turn it back over to Richard to talk to us a little bit about initiatives ongoing with the CF Foundation to expand genetic counselor access for the CF community.
Thanks, Kim. I just wanted to let the audience know about an ongoing project supported by the CF Foundation, which is evaluating the feasibility of using telehealth for genetic counseling. So as we've heard, many families may not receive this counseling, and there are many reasons for that not happening. But one major one is just the unavailability of genetic counselors either in the center or at the healthcare facility. So we are studying, we have gathered stakeholders who would be involved in this process. We have only preliminary information now through some focus groups, but it's clear to us from talking to parents and genetic counselors that without genetic counseling, people sometimes they have to find their own resources, and those may not always be the best resources. So genetic counseling is really important to provide accurate and appropriate information to parents. Some of the barriers that we heard about were concerns about insurance coverage for meeting with counselors, but surprisingly, one of the biggest barriers was just that parents did not even know that genetic counselors existed. And if they weren't available at the center and the parents didn't, ask for that help, it didn't become accessible to them. So stay tuned to hear more about this project. Again, our plan is to develop a service, hopefully through the CF Foundation, that would help overcome some accessibility issues and fill in gaps where counseling resources aren't available, and also to be able to provide genetic counselors who have expertise in CF and can provide the most up-to-date and accurate information to families.
Thanks so much, Richard and Kimberly, for all your contributions on this subject and being here with us today. We're just about out of time, so I want to wrap up. Thank you to our speakers for sharing your knowledge and insight about CF genes building blocks for you and your family. And thanks to all of you for your attention. We are going to take a quick 5-minute break, and then we'll move into our extended Q&A where you can meet and interact with the speakers from today's information session. During this time, attendees are encouraged to click share audio and video and come on camera and ask your questions directly to our speakers. If you would rather ask your questions via the Q&A box, you're welcome to do that instead under the sessions tab on the right-hand side of your screen. We'll be taking questions one at a time in order of request. So we'll see everyone back here. We're a little bit short on time, so make sure you're coming back within the 5 minutes so we can get everybody in tonight. Thank you.